Welcome to the deep dive. Today we're tackling something huge, a massive new resource aimed at understanding the functional side of cancer. For years, big cancer projects like TCGA have focused mostly on genomics, right? Cataloging all the DNA mutations, which is of course essential stuff. But it kind of leaves a gap, doesn't it? How do those DNA changes actually, well, do things in the cell?
Oh, absolutely. It's a critical gap. Think of it like this. Genomics gives you the blueprint for a house, but the proteins, they're the builders, the electricians, the plumbers doing the actual work. They handle metabolism, signaling, building structures, basically all the cell's functions.
And crucially, when we talk about cancer drugs, we're almost always targeting those workers, the proteins, not the blueprint itself.
Exactly. So if we really want precision medicine, we need to see what those workers are up to. We need a global view that's close to the actual function. And that really is what today's deep dive is all about. The Pan-Cancer Proteome Atlas, or TPCPA.
Okay, TPCPA, let's break that down. What exactly did they build here? What is this atlas?
So it's basically a huge inventory of proteins created using mass spectrometry, a really high-throughput method. They managed to quantify almost 10,000 distinct proteins, 9,670 to be exact.
Wow, nearly 10,000?
Yeah, and they did this across nearly 1,000 primary tumor samples, 999 samples.
Okay, that's immense scale. 1,000 tumors mapped at the protein level. What kinds of cancers are we talking about?
It covers 22 different cancer types, 18 solid tumors and 4 liquid ones like leukemias. And the mission, is pretty clear. Use this functional map to understand cancer better, find new druggable targets, and identify really robust biomarkers for the clinic.
Right, the practical applications. So let's get into the how. Mapping proteins across one cancer type is already tough. Doing it for 22 across different labs. That sounds like a recipe for, well, inconsistent data. How did they overcome that? What was the tech breakthrough?
Yeah, that's always been the challenge with large-scale proteomics. The key here was using a specific technique called data-independent acquisition mass spectrometry, DIAMS.
DIAMS.
The important thing isn't necessarily the jargon, but the result. Older methods, things like TMT or antibody arrays, often gave slightly different results lab to lab, made integrating data a nightmare. The DIAMS, it's incredibly reproducible. That meant the 4 different labs working on this could generate data that was directly comparable, but consistent.
So that reproducibility across labs was the secret sauce.
Absolutely. It's the foundation that makes a pan-cancer proteome atlas like this even possible.
Okay, so they got consistent data across 1,000 samples, 22 cancer types, 4 labs.
Yeah.
Impressive. Once they had this giant map, how did they check if it was, actually accurate? Did it reflect what we already know about these cancers?
Great question. That was the first big test. They did what's called an unsupervised analysis. Basically, they threw all the protein data into an algorithm, like without telling it which sample was which cancer type, and the data just sorted itself out. The samples clustered almost perfectly by their known cancer type.
Really? Just based on the proteins?
Yeah. It even clearly separated the solid tumors from the liquid blood cancers, which is strong evidence that they were seeing real biological differences, not just, weird effects from how samples were processed in different labs.
Okay, that's convincing. The map reflects the known diagnoses.
Did it also confirm some of the basic biology we expect to see in these cancers?
It did. They used another technique, SSGSEA, to look for known cancer hallmarks at the protein level. And they found them. For instance, prostate cancers showed high activity in the androgen response pathway, totally expected, as that drives their growth.
Right.
Liver cancers were characterized by proteins involved in bile acid and fatty acid metabolism, again, known liver function. And in cancers like pancreatic cancer, known for being aggressive, they saw high enrichment for proteins involved in EMT epithelial mesenchymal transition. That's the pathway linked to cells becoming mobile and spreading. So it confirms known biology, validates the data. Okay, let's pause here for a sec. We've established the TPCPA is a massive, validated map of cancer proteins. Now, for the listener, you might be thinking, okay, interesting map, but how does this actually lead to better treatments? How do we use this?
Right, the translation step. That's where it gets really exciting. The atlas points directly to new therapeutic avenues. Let's take the first example, targeting E3 ubiquitin ligases with something called PROTACs.
PROTACs. Okay, sounds very sci-fi. Can you paint a picture for us? What does a PROTAC actually do?
Sure, so think of E3 ligases as the cell's built-in garbage disposal system for unwanted proteins. A PROTAC, that stands for proteolysis targeting chimera, is like a molecular matchmaker or maybe a hitman. It's a small molecule with two ends.
Two ends, okay.
One end grabs onto the cancer-causing protein, the target we want to get rid of. The other end grabs onto one of the cell's own E3 ligase garbage disposal units.
So it physically links the bad protein to the disposal unit.
Exactly. It forces the cell to tag the bad protein for destruction using its own machinery. It's basically hijacking the cell's quality control.
Clever. So how does the TPCPA Atlas help with that?
It tells us which E3 ligases are highly abundant, highly expressed in which specific cancer types. See, there are hundreds of E3 ligases. So if the atlas shows, say, that an E3 ligase called HRC5 is really abundant in esophageal cancer, or RNF5 is abundant in liver cancer.
Then you know you have a good handle to grab onto in those specific cancers.
Precisely. It lets researchers design PROTACs that are highly specific to the cancer cells because they rely on an E3 ligase that's abundant there, potentially sparing healthy cells where that ligase might be less active. Very targeted.
That's fascinating. Directly connecting the protein landscape to a druggable strategy.
Okay.
Okay, let's zoom out again. They also use network analysis, right? The WGCNA.
Yeah.
What did that show?
Right. Weighted gene co-expression network analysis, WGCNA. It's like a statistical detective looking for cliques or gangs of proteins. It finds groups of proteins whose expression levels go up and down together across all the samples. The idea is if they move together, they probably work together functionally.
So it reveals functional pathways or modules.
Exactly. They found 13 distinct modules, these sort of functional protein communities.
Can you give an example, one that led to maybe a new insight?
Yeah, let's look at Module 5. This one was highly active, almost exclusively in colon cancers, and functionally it was linked to cell adhesion. The key hub proteins in this module, the sort of central players, included enzymes like GFPT1. These are involved in glycosylation, adding sugars to proteins.
Okay, glycosylation in colon cancer. What's the link?
Well, the analysis showed a functional connection between this glycosylation pathway driven by GFPT1 and the activity of beta-catenin.
Beta-catenin, that's the classic culprit in colon cancer, part of the WNT pathway.
Exactly. So this isn't just saying WNT is important. It's showing at the protein level a potential mechanism how metabolism, specifically glycosylation, might be regulating barcatenin's function in these tumors. That's a deeper level of understanding.
That is deep. Seeing the functional regulation. What about other modules?
Well, Module 6 was interesting, too. It linked to cell stress responses and the cell's powerhouses, and also mitophagy, which is like mitochondrial recycling. The hub proteins there included things like HSP90, heat shock proteins.
HSP90 inhibitors are already used or at least tested as cancer drugs. So
They are. So finding HSP90 as a central player across many cancers in this analysis kind of reinforces that it's a genuinely important, broadly applicable target or vulnerability. It validates that approach at the protein level.
Okay, so we have potential new drug targets like specific E3 ligases and a deeper understanding of pathways like WNT regulation and stress responses. What about using the atlas for diagnostics, for biomarkers?
Definitely. They did supervised analyses too this time. They told the computer which samples were which cancer type and asked it to find the proteins most specific to each one. It identified the top 25 most enriched proteins for each cancer. And importantly, this list included many well-known, clinically used biomarkers.
Like what?
Things like NAPSA for lung adenocarcinoma, CEACAM5 for colorectal cancer, FOLH1 for prostate cancer, MPO for AML, a type of leukemia. Finding these known markers really boosted confidence in the data.
Right, it shows the atlas is picking up the correct signals. Did it find anything new or unexpected?
It did, particularly for brain cancers like high-grade gliomas. It flagged several proteins, ADGRL3, CPNE5, GPR37L1, RGMA, that were highly enriched there but hadn't really been strongly associated with glioma before in large-scale studies.
So potential new diagnostic leads for brain tumors. That's valuable.
Yeah, it gives researchers new avenues to explore immediately.
Let's connect this back to a real clinical problem. What about metastasis of unknown primary, or MUP? That's when cancer has spread, but doctors can't find where it started. It makes treatment really difficult. Can TPCPA help there?
Huge problem. And yes, the Atlas offers a potentially powerful solution. The researchers used the protein data to build a machine learning model, an AI classifier.
An AI to identify the origin.
Exactly. They trained it using 75 key proteins selected from those top enriched markers across 17 solid tumor types. The idea is the unique protein signature of the metastasis can reveal its original tissue type.
Okay, that sounds promising. How well did it actually work?
Remarkably well. They tested it on several independent groups of patients, including people with metastatic ovarian and colorectal cancer. The classifier achieved really high accuracy in AUC, which is a measure of prediction power, between 0.97 and 1.0.
Wait, 1.0 is perfect prediction, right?
Pretty much. It means this protein-based classifier was incredibly good at identifying the tissue of origin for these metastatic tumors. That could be a genuine game changer for managing MUP patients.
Wow, okay, that's incredibly impactful. Now, one more big area of the immune system. We know immunotherapy success depends on whether a tumor is hot or cold. Did the protein data shed light on the immune landscape?
Yes, they used computational methods, like the estimate algorithm, on the protein data to score the immune infiltration in each tumor. And it basically confirmed the clinical reality we see. The non-solid cancers, the leukemias, had the highest immune scores as expected. Among solid tumors, melanoma came out as hot, lots of immune cells, which fits with why immunotherapy works relatively well there.
And the cold tumors?
Prostate cancer, brain gliomas, ovarian cancer, they all showed low immune scores in the protein data. This aligns perfectly with why these cancers often respond poorly to current immune checkpoint inhibitor drugs. It's protein-level validation of that hot versus cold concept.
Okay, so the Atlas confirms known targets, finds new ones, provides diagnostic tools like the MUP classifier, and validates the immune landscape. Let's do our deepest dive now, focusing on one cancer, colorectal cancer, CRC. CRC has this established classification system, right? The consensus molecular subtypes, CMS 1 to 4. But that's based on RNA, on gene expression. How did the protein data line up with those CMS subtypes?
That's a key question they dug into. They had about 191 CRC samples in the Atlas that already had CMS classifications. When they looked at the proteins, they could largely see the same four groups emerge. CMS1 looked immunologically active at the protein level. CMS2 was proliferative. CMS3 showed metabolic protein signatures. And CMS4 had protein patterns linked to that mesenchymal EMT-like state.
So the proteome broadly agrees with the transcriptome.
It does, and they even found some new protein-level details within those subtypes, like the MTORC1 signaling pathway seemed particularly active in the CMS1 immune subtype, and CMS3, the metabolic one, showed enrichment for peroxisome function and protein secretion pathways. Little nuances revealed by the proteins.
And could they actually classify the CMS subtype using only protein data?
Yes, they developed a classifier using just 52 proteins measured by their DIA-MS method. When they tested it on a separate set of CRC samples, it got about 72% accuracy in assigning the correct CMS subtype.
Okay, so you could potentially classify tumors using proteins, maybe useful if you don't have good RNA data.
Exactly. It provides an alternative way to get that classification.
But here's where it gets really interesting. You mentioned the immune part earlier. They didn't just stop at CMS subtypes, right? They looked specifically at immune protein signatures in CRC.
Correct. They did another clustering analysis, this time focusing only on immune-related proteins. This grouped the CRC tumors into two immune consensus clusters. Let's call them CC1 and CC2. And these immune clusters did align somewhat with the CMS groups. CC1 tended to overlap with the CMS2 and CMS3 subtypes, while CC2 overlapped more with CMS1 and CMS4.
Okay, so there's some relationship. But what happened when they looked at patient outcomes, at survival? This feels like the crucial point.
This is the kicker. They had survival data for one of the cohorts. When they compared how well the standard RNA-based CMS classification predicted survival versus how well their new protein-based immune cluster classification predicted survival.
Yes.
The immune consensus cluster classification was significantly better at predicting relapse-free survival, RFS. That's how long patients stayed cancer-free after treatment.
Whoa, wait, let me make sure I heard that right. The classification based purely on the protein immune signature was a better predictor of patient outcome than the established, widely used RNA-based CMS system.
That's exactly what they found. Patients in Immune Cluster 1, which was enriched for CMS23 and had higher levels of activated CD8 plus T cells based on the protein data, had significantly longer RFS than patients in Immune Cluster 2, enriched for CMS14.
That's huge. It suggests that the functional state of the immune environment, which you can measure directly with proteins, might be more prognostically relevant than the overall molecular subtype defined by RNA.
Precisely. It points toward the idea that maybe we've been putting a bit too much emphasis on the RNA classification for prognosis, and that looking directly at the protein-level immune landscape could give us a much clearer picture of who is likely to do well and who might relapse.
That really underscores the power of looking at the functional level. Okay, let's try to synthesize this. This TPCPA project isn't just a massive data set. It's providing truly actionable insights.
Absolutely. From new therapeutic angles like targeting specific E3 ligases for PROTACs to powerful diagnostic tools like that MUP classifier, and now potentially more accurate prognostic markers based on the immune proteome in CRC. It shows high-throughput proteomics is really ready for clinical translation.
And you mentioned this data is publicly available?
Yes, the TPCPA data portal is online. Researchers, clinicians, anyone can go and explore this functional map right now. It's a resource for the whole community.
Incredible. So we've seen the potential for new targets, new diagnostics, and maybe even a better way to predict outcomes, especially highlighted by that CRC immune finding. This leads us to a really provocative final thought for you, the listener, to consider. The study showed protein-level immune classification beat RNA-based CMS for predicting survival in CRC. So the question is, if the functional protein immune environment in a tumor is a stronger driver of patient survival than the broader molecular subtype we usually measure with RNA, are we currently underestimating how valuable real-time protein profiling could be for making crucial decisions about immunotherapy and predicting patient trajectories?
That's the million-dollar question, isn't it? Answering that could really shape the future of precision oncology.
Definitely something to mull over. Join us next time for another deep dive.